Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, I hope all of you are fine and safe at your home. We will start with cloning vectors. Cloning vectors are the DNA molecules that are used as a transporter. Means they are used to transport cloned sequences between biological hosts and the test tube, because we are performing all the experiments in the test tube or in the lab, and then we transfer the recombinant DNA molecule into the biological host with the help of the cloning vector. So cloning vectors are acting as a transporter. They are involved in transporting our gene of interest into the host genome. For example, this is, if I say, is a cloning vector, and we have inserted our gene of interest in it. This is our gene of interest which we have inserted. The vector, when it is transformed into the biological host, it will transfer this sequence into the host genome. So it is acting as a transporter or as a carrier molecule. Any molecule or any vector which possesses these four properties, it can be used as a cloning vector. Like, these are the properties that a cloning vector, every cloning vector should possess, and these four properties are very important or essential for a vector to perform the function of a cloning vector. First one is its ability to promote autonomous replication. Autonomous replication means it should be able to replicate by its own. It does not depend upon the host organism for its replication. If you recall your molecular biology course or any such kind of course that you have studied during your BS or MSc program, you have studied about the replication. It involves origin of replication. If I give you an example of the E. coli, then it has an origin of replication which is known as locus. It is known as locus. So this is the origin of replication which is important for the start of the replication process. So the molecules which possess their origin of replication, they are known as autonomous molecules. Like, they should be able to start the process of replication by its own. Therefore, the cloning vector should possess this ability that they should promote the autonomous replication, that when introduced into the host organism, they can replicate by its own and they can produce their multiple copies within the host organism. Second important property is they contain a genetic marker usually dominant for selection. Genetic marker that is mostly used, it is an antibiotic resistance gene which is mostly used. It is antibiotic resistance gene. For example, the bacteria that we are using as a host, it cannot survive in a specific antibiotic condition if we provide it into the media. That's why if we introduce a plasmid which contains that antibiotic resistance gene in it, then if the bacteria survive, it means that the vector has been transformed into the bacteria. So it is used as a genetic marker. I will write it as antibiotic resistance. We will discuss in detail shortly about these properties. Then the third important property is unique restriction site to facilitate the cloning of gene of interest that we have that we are going to insert into it. Restriction sites provide cutting sites for the restriction enzymes. These are restriction enzyme cutting sites, and they provide the unique restriction sites because before starting the process of cloning, we must assure that the enzymes that we are going to select, those enzymes do not make any cut within the gene sequence, because if they cut the gene sequence, then our gene will not be cloned as a whole gene. For example, this is the insulin gene, and if I select the two restriction enzymes, one of which is the EcoR1, suppose, and I didn't check whether EcoR1 cut within the gene sequence or not. And when I incubate the PCR product with the EcoR1, in addition to making the cut at the end, it can also cut within the gene sequence if its cutting site is present within the gene. Then, as my purpose is to clone this full fragment, but now instead of full fragment, I will get a clone of only this part, that is partial fragment or the partial gene of the insulin. So to avoid this purpose, we need to select that restriction enzyme that do not make any cut within the gene of interest. Therefore, the unique restriction sites are introduced into the cloning vectors that rarely contain any cutting site within your gene of interest. The fourth important property is about the size of the cloning vector. That the cloning vector should contain minimum amount of the non-essential DNA to optimize the cloning, because if more non-essential DNA is present, then it means that the size of the vector is large and there is a particular capacity of every vector to carry a particular size fragment. There are different types of vectors that are shown on this slide and we will discuss shortly. So every vector has different amount of has the capacity to incorporate the different size of the DNA fragment. So if the cloning vector contain less non-essential DNA, it means it can carry the large fragment size. Mostly the types of the cloning vectors that are used in biotechnology, there are 5 types of the cloning vectors. These are known as the plasmids or the phage vectors and the cosmid vectors, or the BAC, which is also known as the bacterial artificial chromosomes, or the YAC, which is also known as the yeast artificial chromosome. So these are from the basics towards more advanced types of the vectors that are used in recombinant DNA technology technique. So first we will talk about the plasmid. We know that plasmids are extra chromosomal circular DNA molecules that can autonomously replicate inside the bacterial cell, and we know that this is the property of the replication vector, that the plasmid should be able to autonomously replicate within the bacterial cell. And now it has a cloning limit. Cloning limit means the size of the DNA fragment that can be cloned into the vector. It has that limitation. For instance, it can carry 100 base pair to 10,000 base pair size, or 0.1 kb to 10 kb. It is same. If we talk about the base pair, then we multiply kb with 1000, and if we are converting the base pair to kb, then we will divide it by 1000. So the limit of the plasmid is 0.1 to 10 kilo base pair fragment that can be inserted into the plasmid. This is its cloning limit that we were discussing at the fourth point. And these are understood that this plasmid will contain the unique restriction site as well as it contain a selective gene which act as a genetic marker, which is the second important point. Then the second kind of vector that is being used in recombinant DNA technology, it is known as the phage viruses. Phage viruses are actually derivatives of bacteriophage lambda, and we know that bacteriophage lambda are the bacteriophage or phage. These are the bacterial viruses that use E. coli as a host organism, and we know about the structure of the virus, that it contains a head, it contains a tail, and it contains a tail fiber. So it is consists of mainly 3 components. Then it is a linear DNA molecule whose region can be replaced with a foreign gene without disrupting its life cycle. We know that the virus, when make entry into the host cell, it then carry out its all the processes by using the host resources. So when the lambda tail fibers, these fibers absorb to the cell surface receptor, the tail contracts and DNA that is introduced, it is injected. This DNA can be circularized with the help of specific sites which are known as the cos sites. So, and this lambda begins its life cycle into the bacterial host. So, if we introduce any foreign gene into its DNA, then that foreign gene can also be introduced into the host cell when the host cell is transfected with the phages or bacteriophages. And the cloning limit of the phage is more than the plasmid, that it can carry a fragment of 8 to 20 kb. Then the third kind of vectors are the cosmid vectors. Cosmid vectors are the combination of plasmids and phage, like they are prepared by using both plasmids and phage vectors to increase the cloning limit, which is 35 to 50 kb fragment. These are actually plasmids, and in the plasmid, the cos site from the phage viruses has been introduced in it, and the presence of these cos sites within the cosmid vector, it permits the in vitro packaging of cosmid DNA into the lambda particles. The next advanced form of the virus is the bacterial artificial chromosome. Bacterial artificial chromosomes are based on the bacterial mini F plasmid. These are the chimeric DNA molecule which use a naturally occurring low copy number bacterial plasmid, origin of replication, as that present in the mini F plasmid of the bacteria, and their cloning limit is 150 to 300 kb, or at some places 350 kb is also mentioned. The advantage of the bacterial artificial chromosome is that they can be cloned as a plasmid in a bacterial host, and its natural stability, which generally permits the cloning of the large fragments of the DNA. More advanced form that can be used within the eukaryotic host organism, it is the yeast artificial chromosome. Yeast artificial chromosomes, they are the artificial chromosomes that contain the sequences which are similar to the sequences of the eukaryotic chromosomes, like they contain the telomere sequences, they contain the origin of replication, they contain a centromere region, and they contain a selectable marker which can be used for identification of yeast cell. So these are the cloning vehicles that can propagate in eukaryotic host as eukaryotic chromosome, and they can be used to clone very large insert of 100 to 2000 kb, or in some book 3000 kb fragments can be cloned by using the yeast artificial chromosome. These vehicles are also the plasmids, and they are the chimeric DNA because they contain the telomere region, and as well as they contain the yeast centromere region, and they can be used as a vector in both eukaryotic as well as the bacterial genome. We will discuss one example of the cloning vector which is most commonly employed. These are the plasmid vectors. You can see the structure of the plasmid vectors. Sorry, plasmid vectors are usually 1.2 to 3 kb in size, and they contain all the important properties that a vector should possess. For instance, they should have the origin of replication, They contain a gene that can permit the selection process. In this case, that selective gene, which is ampicillin, and R is for resistance. It provides resistance to ampicillin and it is encoded by a gene which is known as beta lactamase. So beta lactamase is a gene which give resistance to ampicillin antibiotic. So when this plasmid is transformed into the bacteria, if the bacteria is susceptible to the ampicillin, and when it is transformed with plasmid, it will get resistance from the ampicillin due to the presence of this beta lactamase gene. This lactamase gene will inactivate the ampicillin which is provided in the media. Then you can see this bracketed region which is in green color from this position to this one. This is the region in which the exogenous DNA or your DNA of interest can be inserted, and this plasmid does not contain extra region which is non-essential DNA region. So these are the main properties of the plasmid vectors that we will also discuss in detail shortly. One important characteristic of the plasmid is the selective marker. As I told you earlier that ampicillin, it is actually ampicillin resistance gene and it is used as a selective marker. It is required for the maintenance of plasmid in the cell. We will come to know that the plasmid is present in the cell due to the presence of this selective marker. Because of the presence of the selective marker, the plasmid will become useful for the host cell, because as I give you earlier example that if the host is susceptible to ampicillin, now it will become resistant due to this plasmid. So it is beneficial for the host cell. And under the selective conditions, only cells that contain plasmids with selectable marker can survive. Selective condition means if we are providing within the media ampicillin. If you have studied the course of microbiology and you have performed some experiments, you may know that we take a Petri plate in which we put the media, which is mostly for bacterial growth. We use the Loria Bertini media, which is also known as LB media, and on this LB media we generally do the streaking of the colonies, if you recall, and next day we observe that at the place that we have created we see many bacterial colonies, and this is the simple media. But under selective conditions, selective condition means within the LB media we also add ampicillin, which is an antibiotic, and we want to check whether our host cell is sensitive to the ampicillin or it is resistant to the ampicillin. So for that purpose, within the LB medium we have added the appropriate concentration of the ampicillin antibiotic. And now what will happen when we streak the bacteria on the plate? Then only few colonies will appear. These colonies are those colonies or those bacteria which contain any plasmid which have the ampicillin resistant gene. Only those bacteria survive while all the other bacteria does not survive. So for that purpose, for selective marker, we use the genes that provide resistance to several antibiotics. For example, here we have seen the example of the ampicillin, the resistant gene. Other examples are like we can use the genes that can make resistant to the antibiotic like neomycin or the chloramphenicol. So these antibiotics are most commonly used antibiotics whose resistant genes are present within the plasmid cells. We have also said that a bacteria or bacterial plasmid that we are going to introduce into the bacteria, it should possess the origin of replication, so it can autonomously replicate. What is meant by origin of replication? Origin of replication is a DNA segment, as you can see here, which is recognized by the cellular DNA replication enzyme, which is DNA polymerase. So origin of replication, this region has specific characteristics that allow it to act as a start point for the process of replication, and the DNA polymerase will recognize this region and then will start the process of replication of the vector from here, and then we get, it will replicate the whole enzyme, whole plasmid, and then we get multiple copies of this plasmid within the host organism. If this region, which is origin of replication, if this region is not present within the within the plasmid that we are using as a vector, then this DNA will not be able to replicate within the host cell. So it is essential component of the plasmid. Another important is the region where we introduce the foreign gene. That region contains unique restriction sites, and these unique restriction sites, they are located in a special region which is known as polylinker region, or we also call it as the MCS. MCS stands for multiple cloning site. Multiple cloning site means here the cloning sites for more than one restriction enzyme are present. So these are the cloning sites which are present in the cloning vectors. It is actually a segment of DNA with several unique sites for restriction endonucleases which are located next after the other. Like after BAMH1 there will be the restriction site for HIN3.1, and after that there is SOL1. So there is one after another. These restriction sites are arranged in such way. These are the unique restriction enzymes. Maybe you are familiar with some of them, like BAMH1 is very common, EcoR1 is very common, SMA1 is very common. These are the common and unique restriction enzymes whose restriction sites are present in the polylinker region. These restriction sites of the polylinker region, they are not present elsewhere in the plasmid. In the other whole plasmid, you cannot see any other restriction site. We only use this polylinker region for insertion of the foreign gene into this length. For example, if we choose the BAMH1, which is located at this position, and KPA1 over this position, then we will treat the plasmid as well as our cloned gene with these restriction enzymes, and we have introduced the restriction enzyme sites during the PCR amplification with the primers. So our foreign gene will get inserted into it, and all this region of the restriction sites, it will get removed or it will be replaced with the foreign gene. Cutting the plasmid with one of the restriction enzyme that can recognize a site in this region, it will not disrupt any other feature of the vector, because if we are treating it with BAMH1, it will make a cut over here, and all the other features of the plasmid, they are not get disturbed. There will be intact origin of replication, there will be intact ampicillin resistant gene. Or if we are treating it with more than one enzyme, like with the BAMH1, as well as we are treating it with small one, then what will happen? It will make a cut over here and here. This fragment will be lost, and this is a fragment which only contains the restriction site. So this is not affecting the other properties of the plasmid, and all the other properties of the plasmid will remain intact. So here is the example that how the multiple cloning site help to clone a foreign gene into the vector. As you can see, this is the gene of interest and this is the vector. Suppose we have amplified this gene of interest with the PCR, and we have introduced only one enzyme site, which is the EcoR1 at the both ends of this vector, and this is the overhang of the EcoR1. So when we treat this with the EcoR1, then this part will get removed, this part will also get removed, and we get this specific sequence. Similarly, to introduce it into the vector, we need to cut the vector with the EcoR1, and you can see here is the cutting site of the EcoR1. So we will treat it with one enzyme only. It will make it a single cut in it, and because of this single cut, this circular molecule will become linear, that it become open, and you can see this open end. Now it contains the overhangs, and these overhangs we will discuss when we will talk about the restriction enzymes, that they produce the sticky ends or the blunt end. Sticky ends means they can stick to their counterparts, and their counterparts are present in this gene sequence that we have cut it with the EcoR1. So when we mix it with it, we use a special enzyme which is known as the ligase enzyme. Ligase enzyme will join this fragment into this space, and in this way we get the recombinant plasmid which contain our gene of interest at this position, where we have make a cut in the enzyme, and all the other properties of the plasmid, they are intact. So here is the example, like this is the sequence of the polylinker, like how the cutting sites of the plasmids are present. They are one after another, as I told you before. As you can see, this is the restriction site of the EcoR1 at this point, GAATTC. Then after EcoR1, there is cutting site for the SAC1, and just after the SAC1 there is cutting site of the KPN1, and within the KPN1 there is the cutting site starts for the small one. So these cutting sites can overlap with one another, and we use the enzymes which should not overlap, like we cannot use SMA1 and KPN1 at the same time. We will use SMA1 and the SAC1, or the SMA1 with the EcoR1, or the BAMH1 with the SAC1. We can take any combination of the enzymes. So this is the cutting site of the BAMH1, which is the GGATCC, then XBA1 and SOL1. So these all are the restriction sites, so restriction enzyme sequences which are present in the polylinker or this region. This is the origin of replication, which is necessary for carrying out the replication of the vector. This is the ampicillin resistant gene, and this is the polylinker region where the gene of interest can be inserted. So if we treat it with the EcoR1 enzyme, it will make such kind of cut. Here you can see the formation of the sticky ends. Sticky ends are like overhangs, like this is, for instance, if it is the 5' end, and this is the 3' end, you can see the 5' end contain only G and all other part is empty. And similarly the opposite strain also contain the G and all other part is empty. This part will be provided by the foreign gene. When we treat the foreign gene with the EcoR1, such kind of ends are produced because this enzyme make a cut after G. This is the cutting site of the EcoR1. It will make a cut over here. And one important thing about the restriction enzyme is that they are the same when we read them in both directions. Like here you can see it is GAATTC, and in the opposite direction if you write it, it will be GAATTC. So these are such kind of sequences are known as the palindromic sequences that we will discuss shortly in the restriction enzyme topic, maybe in tomorrow lecture. They will produce such cuts in the DNA, and now these sticky ends can come and bind with this TTAA, and then we use the DNA ligase enzyme which will ligate this A with the G and this G with the A. So in this way we get the recombinant plasmid. For like we have isolated a genomic DNA, and we will treat the genomic DNA with the EcoR1. It will produce many fragments which contain the sticky ends of the EcoR1, and these fragments can join the cloning vector, and with the help of DNA ligase enzyme which uses ATP as energy source, we get the recombinant plasmid. Sometimes the cloning vectors, in addition to the ampicillin resistance gene, which provide the selection process, another selective gene is also present within which the restriction sites or multiple cloning site is present. That gene is known as the LAC-Z prime. LAC-Z is a gene which encode an enzyme which is beta galactosidase, and beta galactosidase is involved in the metabolism of lactose. If you recall the LAC operon, which consists of three genes, LAC-Z, LAC-Y and LAC-A. So this LAC-Z gene is from the LAC operon. So what is the purpose of this gene? We will discuss on this slide that we have a foreign gene and we want to introduce it into the vector. We digest the vector and gene with the same enzyme. As you can see this arrow, we make a cut in this cloning site that it becomes single stranded. It becomes open, and then we cut the foreign DNA with the same enzyme and then we join it with the help of DNA ligase. Then there are two possibilities. In the one possibility that the fragment does not get inserted into the vector and the vector self-ligate because it is compatible to be ligated with itself, so it will self-ligate. And in the second case, the foreign gene has been incorporated into this, as you can see in the green color. This is all theoretically that we are supposing that our fragment is inserted into it. How we will come to know? We will transform this vector into the E. coli cell and then we will select it on the ampicillin plates, which will contain a substrate of the beta galactosidase, which is X-gall. This is the double selection of the recombinant vectors or the host organisms which contain the recombinant vector. Now you will see two types of colonies. If I draw a plate, you will see the blue colonies of the bacteria as well as the white colonies. As you can see, this is white, so in place of white I will encircle it with the purple color, that these are the white colonies. So what is meant by these blue colonies and the white colonies? Blue colonies are due to the metabolization of the X-gall. It means that this LAC-Z enzyme is active. Why this LAC-Z enzyme is active? Because the foreign DNA fragment is not inserted into it and it has not disrupted its function. The function of the beta galactosidase gene will get disrupted when a foreign fragment is inserted into it, because it is an extra fragment and it will disturb the protein formation which is encoded by the LAC-Z gene. So beta galactosidase will not be formed. Beta galactosidase will become inactive and the transformates will be white in color, as you can see in this diagram. And if you see the blue vectors, blue vector means these are the transformates in which the beta galactosidase is active and it is metabolizing the X-gall and giving the blue color. So this process is the double selection, which is on the ampicillin plate. Only those colonies will grow which contain the plasmid, and that plasmid could be an empty plasmid or with a sequence. So for that purpose we have used the LAC-Z selection process, in which we have introduced the X-gall, which is a substrate of the LAC-Z or the beta galactosidase enzyme. When it will metabolize it, it will convert it into the blue color product. So blue colonies will appear, and if white colonies appear, those white colonies shows that the foreign DNA fragment has been incorporated into it. So dear students, this was all about today's lecture, in which we have started from the introduction of the cloning vectors, and then we have discussed the properties of the cloning vectors, then what kind of cloning vectors are used, and in detail we study about the plasmids, that they possess all the properties that a good cloning vector should have. And then we discussed the example, how can we use the double selection for identifying or screening those colonies of the bacteria which contain the recombinant plasmid having our gene of interest. So this was all from my side. If you have any question, please feel free to ask. Thank you.